It's amazing that explaining life's immense diversity all comes down to some genetics and some biochemistry. And life on Earth is just one family. And what's true for you is true for all biology.
Hello, welcome to Genetic Shambles, our fortnightly show where we speak to geneticists about all manner of different things. We've obviously spoken quite a few times about COVID, and we've talked about Neanderthal genes, and today we're talking specifically to one of, someone who introduced me to Dublin's wonderful Dead Zoo. We may well talk about the Dead Zoo and how genetics may have changed the way that taxidermists actually now stuffed a kangaroo that they'd received. There may be many different things we talk about in terms of the relationship between taxidermy and genes today, but that might not come up either. So thanks very much. This is kind of the penultimate one, but it's not. In one way, it is. Next week, we're joined for the final with a conversation with Steve Jones, who, as anyone who knows Steve Jones will know, is one of the top 5 experts on snails, but is also one of the most wonderful writers about genetics and also about Charles Darwin and about many other things as well. And then we'll also be having a little bit of a Genetic Shambles on the 12th of December during our 24-hour science show, which you can find out more about that on cosmicshambles.com. We're basically doing a live 24-hour show. It would have been the kind of thing that we dot around theatres, but instead we're just going to do it in one fell, possibly extremely destructive for me physically swoop. So a few things about Genetic Shambles. First of all, thanks to Genetic Society and Milner Centre for Evolution at the University of Bath, who are the reason that we are making Genetic Shambles. And also, if you have any questions tonight, you can just pop them into the live chat or go to Cosmic Shambles which is on Twitter, and Trent, our producer, will make sure that I see those questions. So if you've got any live questions at all, we will be dealing with those. And that's pretty much all you need to know. So let me tell you about Aoife McLysaght, who I think I first met doing a gig in Dublin in Trinity, and I can't remember if she was the reason that I was able to get into their fantastic Natural History Museum, which is not the Dead Zoo. The Trinity Natural History Museum is another wonderful thing. She's a professor in molecular evolution in the Molecular Evolution Laboratory of the Smurfit Institute of Genetics at the University of Dublin, Trinity College, and was also delivered the 2018 Christmas lectures with Alice Roberts as well, which was brilliant. And so we're just going to hello, Aoife, how are you?
I'm very well, thanks, Robin. Thanks for the lovely introduction. I remember meeting you very well.
You did introduce me to the Dead Zoo, didn't you? It was
that, but I remember. So you were over. You did an event in the Science Gallery in Dublin, and I was lucky enough to be invited along to the dinner afterwards with you. And I remember. I don't know if I'm going to embarrass you now, but we were all at the table chatting, and you were talking about doing monkey cage and all that kind of thing. And you said, "Oh yeah, we seem to have terrible trouble finding female guests." And then you looked at me and you were like, "You appear to be female." I think you invited me onto monkey cage shortly after that, so that was fun.
Yeah, I think that was in Cheltenham Science Festival. I think was the first one. But
that was great.
That was
baptism of fire.
It's amazing how scared kind of people feel about the fact. It's quite a relaxed chat, but people always presume that Brian's going to somehow suddenly go into theoretical physics and destroy the fabric of space-time around us. So let's talk about, first of all, your career. Now, I remember speaking to you a while ago, and you saying that it wasn't as if you were into biology and ideas of evolution from an early age. There was kind of almost a Damascene moment that you had a great teacher, and suddenly it sounds to me like everything just clicked, and you went, "This is a fabulous idea."
No, I had, so I don't remember being that into science when I was young. I mean, so when I say young teen or in primary school, that kind of thing, I generally liked everything. I liked school, but I had this fabulous teacher, and was quite unusual because he had a PhD, which is a bit unusual for a school teacher. And he always went a little bit beyond. And I can distinctly remember when he did genetics with us. And one of the things he did was, so I described the way he used to teach as being actually very like what you need to do when you're really doing science for real. So the misconception about what happens when you're doing science is that there's this stuff you have to learn, and when you know it all, then you are coronated as a scientist. But instead, it's a process, of course. It's figuring things out, it's noticing stuff. And he managed to somehow replicate that in the classroom. And the example that I remember really distinctly was when he was doing genetics. So he told us about, you've got the 4 bases and they're represented by the letters A, C, T, G. And he was talking about, there's a gene. And every time he was mentioning a gene, he would always just do A, T, G and then do a squiggle. But he never pointed out the fact that he was writing A, T, G and not just any other 3-letter combinations of that 4-letter alphabet. He just always did it. And I remember just noticing that and going, he always starts with ATG. I wonder, why does he always start with ATG? And it was later on then that I learned, actually much later on, that I learned that this is a special 3-letter combination, which is called the start codon, and it's a signal for the start of a gene. And it was just like he just left it there for us to notice and to make our own little discovery in this stupid little way, something that was already known, but we had that little experience of noticing something, going, there's a bit of a pattern here. They always start with ATG. And so that got me interested in just the idea of figuring things out. I discovered that I like looking at things and figuring them out. And then, and also just the thing I really, really liked about genetics was that it's so broad and it applies to so widely to biology, and that it's not, one of the things that in our course at the time, so I'm not saying this is a criticism of biology, but in our course at the time, most of the biology we did was remembering things, like drawing pictures with the correct labels on them. So you draw a picture of the eye and you had to label the lens and the retina and the blah. And that was very boring. But genetics was about understanding things and getting the ideas and getting the principles and then seeing how this applies. And of course, being genetics, it is the language of all life. So once you understand the way genetics works, you can use that understanding and apply it in any place, any part of life. So I just really, really enjoyed that. I enjoyed the figuring things out bit, and I enjoyed how genetics was a really special angle, a really powerful angle into figuring things out. So yeah, it just got me excited.
Should we be learning about genetics from an earlier age at school? I just wondered, both perhaps in biology and in physics, for instance, most people go through most of their school life in a Newtonian universe, and it might only be by about the 3rd year that they start to move into an Einsteinian universe. And in the same way, even though obviously, genetics can be very, very complex, that complexity just beginning to throw those seeds, just little ideas. Do you think that would be a good idea?
I think it would be a fabulous idea. I think it makes a lot of things make more sense. I don't think it makes them more confusing. Of course, it's possible to overwhelm somebody with information. So it should be age appropriate in terms of how much understanding you expect on the other side and how much material you expect them to get. But I think I think it's one of these things that genetics has actually entered into popular culture a lot. People know what the word gene roughly means. Like, I don't think you could ask somebody to define it. That's a bit tricky. But they've heard about genes and they've heard about DNA, and they know that it's something to do with what makes you what you are. So you've got a really good starting point, actually, that there's so much there that's already known in popular culture, and young kids know that too. I go into schools quite a bit and I give little talks, and I'm always really, really impressed at how quickly they catch on to the ideas. So I talk to them about, I usually use cats actually as my way of explaining genetics, because with cats, the coat patterning, so the coloring, and that whether it's patchy or solid color and these things, it's just a few genes control that. And it's very straightforward genetics in the sense of not being multiple genes interacting and all these kind of things. It's kind of a simple trait. And so I do this game with them where I show them a picture of our cat, which happens to be a fully black cat. And I tell them I'm going to teach them how to do a magic trick, which is that they can tell me the color of that, the father of that cat, without ever seeing that cat. And then I show them then the litter mates and the mother, and I explain to them how this is enough information to figure out the color of the father, even though they've never seen the father. And so we step through it, and they figure it out. They always figure it out, and it's really great. And that is just that. To do that, I do this with primary school children, and to get the answer, they need to understand the basics of inheritance, so that you have 2 copies of each gene and the way you pass them on. They need to understand the idea of dominance, that some genes are dominant over others. And they also need to, because of the way cats are a bit funny, the coat color genes are on the X chromosome. And when you get a tortoiseshell cat that's got orange and black patches, that's when that cat has on one of her X chromosomes the black allele, the black version of the gene, and the other one has the orange. You got some patches where one is turned on and some patches where the other is turned on. So there's a lot of things happening here. They need to get the basics of all of these ideas, and they figure out that the dad is orange, and it's brilliant and it works. And I think it's something that definitely could be introduced earlier, and it can be fun to do it because then you start seeing it around you. Another time I went into a primary school and I got them to extract DNA. So you can do it with households, kind of quite easy to obtain chemicals, right? So you need quite pure alcohol, which you can get in the pharmacist, so for cleaning wounds. So you can get the surgical spirits essentially. And then you just need dish soap and some salt. So that's what you need to be able to extract DNA. And you can get DNA spooling out onto a little wooden toothpick or something like that, or a cocktail stick. And I did it with strawberries because there's lots of DNA in strawberries. It's easy to get them out, the DNA out, because they're quite mushy. But then you start saying, do you want to extract DNA from something else? They all go home and they start trying to extract DNA from stuff in their kitchen or whatever. And I think you can introduce the ideas. I think I think it's fun. I think they enjoy it. And I think it would probably make a lot of biology make more sense when you think about it from this point of view. But I would also add evolution, because understanding genetics and evolution goes hand in glove. And then it makes biology make a lot more sense.
See, do you know yet why blue-eyed cats always have bad teeth? Now, I don't know if this is true or not, but it was an observation of Darwin's. And of course, Charles Darwin, that was before he knew about genes. But that was one of my favourite things of his observations. He had something about bald dogs. I can't remember what that's linked to. And he said blue-eyed cats always seem to have bad teeth as well. I love that. Again, that joy of observation, which must be so much part of, as you were saying, that observation of that ATG. That immediately means that you definitely found the right path to take, didn't you, if you were noticing those things?
Yeah. Oh, it must have been. I thought you were about to tell me a joke there. I didn't know the blue-eyed cats and the bad teeth, so I don't know the answer to that one. I thought it was set up for a punchline.
Oh no, I'll do the traditional touching the glasses in the old style clubs there. Those blue-eyed cats. That's but I do think that bit of being able to observe it in real life. I remember the comedian Stuart Lee once saying Stuart was adopted, and when he went round to his first girlfriend's house, he suddenly was struck by the fact that all of the family looked quite similar. Mm-hmm. And it's something that he hadn't really thought about from his own. And I know there's a scientist who I won't name, though, long dead, but a scientist who one day had to explain to a girl who did not know that he was actually her father. He thought, I need to broker this. And they were sat on the beach with their just bare feet. And he said, isn't it funny how similar our feet look? And I think, all of those little things, those details, which mean that this is not just laboratory observation, or this is, as you said, you can go into your kitchen, or you can look across to someone else and you can look at the similarities of people on a railway carriage and start to think, you know what, that person looks not that dissimilar to that person. So I'm presuming there's a high possibility we can go back the family tree and before long they entwine. All of those things to me seem to give part of the wonder of our now understanding of how that inheritance works.
Yeah, and then you take that idea and you apply it to animals that you see in the zoo or in the dead zoo. And, you can, that same principle applies. So, yeah, you can take these ordinary everyday observations, which is children look a bit like their parents. And then, you don't realize yet, you can step through these ordinary everyday observations and get to something quite profound, which is maybe, your eyes opening on understanding a bit of evolutionary relationships and the relationships of all the various life on Earth. And so I think, it's incredibly powerful, and I think it's very accessible. And I think it's probably a bit more accessible than Einsteinian physics, which is quite difficult. At least I find it difficult. But I think it's something that you can have all these everyday examples, and you can do lots of fun stuff with it.
You're right about Einsteinian physics. I've spent the day writing a chapter about time in the book that I'm doing at the moment, and at the end of the day, I've just realised I haven't really understood anything I've typed out there. But this is you. I mean, you've done a lot of public engagement, and some of the stuff which is very easy for anyone watching this if you want to see afterwards. A lot of the stuff that you did, for instance, at the electric picnic, where you are able to, and I wonder, finding those stories which sometimes almost appear to be a shaggy dog story, but by the end of it we find out the genes that are needed for that particular shagginess. I mean, that's when do there must be certain stories that you can just see almost imprinting themselves on people as you tell them those stories.
So I don't know that I'm good at guessing what other people like. So what I do is I talk about things that I like. So my strategy to picking things to talk about, if I do electric picnic or when I've done shows with you, I describe it as, I think over the previous year, and I look for the things that made me go, ah. So I was like, that made me go, ah, that's interesting. And so it's something that I find a little bit curious, and I dig around in it. And I think that'd be fun to tell about that, to talk about that. And usually if it made me go, ah, it's because it's often a little bit away from what I'm doing on a day-to-day basis. And so I stumbled upon it and I went, that's weird. How could that be? And I look at it and I meander around. And so then the way I talk about it sometimes then when I'm talking to the public, it kind of describes those meanderings as well, in the sense that I'm kind of saying, it could have been this, it could have been that, but it turns out it's the other. And that's, that's the way that I find things when I decide I want to share a little bit of science. I find that the things that I find that to me, I find really interesting and extraordinary. I just hope that it'll work for other people too.
What have been the most recent ones where, I mean, especially in this situation at the moment where quite often we've not been able to sometimes communicate these ideas and have those chances as live events? There must be some that you think, I just want to share this with as many people as possible.
Oh, you're putting me on the spot here now. I'll tell you later. I'll see if something comes into my head. I'm trying to think if I don't know if I had something really, really recently about that it was the things that made me go, I'm not sure. You're putting me on the spot and I can't think of something now.
What do you think in terms of people who are interested but not scientists? What are the things you find you're most often asked in terms of their hankering to understand the implications?
So you mean understanding bits of science in general? Is that
generally the implications of what we understand and because it is such a rapidly moving discipline at the moment, and it is the kind of thing that occasionally will make the newspapers, and very often when it does, I know that you'll probably be sitting there going, well, they've only copied out the first 2 lines of that particular paper then.
Yeah. Or they've only read the press release. I think the thing that so in terms of understanding implications, I think the thing that people are most interested in tends to be around gene therapies and editing, and of course the Nobel Prize in, I think it was I can't remember, it was in chemistry this year, went to Doudna and Charpentier for their discovery of a mechanism for gene editing. And that's one that did capture the public imagination very quickly, I think. The idea that, you can change genes and you can do it in this really, really targeted way. And I think that's something that's very interesting for people to understand. But I also really fall for I'm really interested in just the intrinsic value of knowledge without it having automatic implications for other things, or maybe not necessarily having implications for people in their lives, but just the pleasure of understanding things. And I find in times when I've given talks about things as apparently obscure as just, well, not obscure, but not necessarily going to have extraordinary implications for people, just in terms of something like how an eye begins to develop, and how the gene that triggers the development of an eye in us is essentially the same gene as does it in mice, and is essentially the same gene as does it in octopus, an extremely distantly related animal. And basically everywhere you find an eye, it's the same gene. So it's descended from our common ancestor. So that doesn't necessarily have immediate implications for people, except perhaps that the big implication, which is it's kind of presenting this evolutionary relationship and the connectedness of all life, presenting it in a very, for me, a very clear and stark way, which I find kind of it's humbling, and at the same time it's enriching, because I think, it's humbling to see this connection between all life on the planet, but somehow there's something that you feel kind of bigger afterwards for having understood it, right? So it's two things happening at the same time. Like, you've understood this thing and you've discovered this thing, so you feel this swell of pride in the knowledge and the understanding, even if you've just understood that, something that about us, which is, as complex as the eye, has these beginnings that are shared across all life. So yeah, these are the kind of things, that's the kind of way I approach things. I enjoy the knowledge, I enjoy the figuring out, and I try to share that.
Well, I remember you once watching a talk where you talked about how far into this discipline you'd gone before you then suddenly had that revelatory moment of going, this incredible connective tissue. When you see what Paul Nurse and his team did, and the fact that, yeast can't sit, as far as we know, in whatever it's contained in at the time, and think, wow, what incredible implications that not merely the code that I have is, but as you said, we can. And that I think is very beautiful. The fact that you can be perhaps observing something so close up that it's not when you're in the laboratory, it's not when you're doing the research, it's when you're just gazing out of the window, it's when you're going for a walk, you suddenly think, oh, this connection is in every living thing that we can observe.
No, so when I had the example you're reminding me there of, is it's literally when it occurred to me in a kind of very it occurred to me very suddenly in a certain way, the fact that the DNA connection of all life to each other is not a metaphor. And that I realized, but actually, no, it's a physical connection. The DNA has touched the DNA of the previous generation, which has touched the DNA of the previous generation, and so on forever, as long as there is life. And that's a very, in a certain way, mundane observation. And you could tell that to somebody on the first day of their genetics or biology. And possibly I was told that on the first day, but it didn't sink in or something. It didn't mean the same thing until I came back to it again later. And that happened because I was talking about genetics to somebody else. And I was, to somebody who wasn't a geneticist, and I was explaining things. And it just dawned on me as I was saying this, I was kind of rattling something off about, DNA connects us all. But then I was like, oh my God, it really does. And it was kind of funny. I was, already a professional geneticist, and I just hadn't thought about it that precise way, that this is more than a metaphor. It's like so I imagined it like a big long chain of people holding hands. So you're not holding hands with everybody in the line, but there's an unbroken chain. So you are linked to the person at the beginning of the line, even if you're not directly holding their hand. And that when it dawned on me like that, it was suddenly, it was like sometimes people describe suddenly understanding infinity or something like that. And so it was a bit like that for me. I was like suddenly seeing that this is a really big, long, unbroken chain, by definition, an unbroken chain, because when the chain breaks, the DNA doesn't get passed on, and that's a dead end in terms of evolution. So anybody who is alive today, by definition, has ancestors in every previous generation. And so there is an unbroken chain. And that was kind of a silly and a profound moment at the same time.
Yeah, because when you say mundane, I think it is so far from mundane. And when you think of those hands, also, if you think that each time you then get a species divide, so as you go back those chains, and as we've talked about on this many times before as well, the fact that it's no longer a tree of life, it's become something far more complicated in what you see. We've had loads of questions coming in, and I'm going to take the first one from Adam Deeks wants to know, what was the first thing to have DNA? Did something not alive, I'm saying it like that, by the way, to show his inverted commas, not alive, mutating something alive, triggering the beginning of evolution?
Okay, so the first part of the answer there is that what we believe, so of course we can't go back in time, but we have, let's say, very good inferences that we're reasonably confident in. We believe that the first living thing didn't use DNA as its genetic material, but used a chemical relative, which is RNA, very, very similar. So DNA is deoxyribonucleic acid, and RNA is just the ribonucleic acid. So you take the deoxy off. So it's a slightly more unstable molecule. But the reason it's considered to probably be the first, the genetic material of the first living thing, is because it both carries information and can be active. So it can do stuff as well, kind of enzymatic. It can control processes in the cell, whereas DNA can only carry the information. So what happens in our cells now, we've got DNA carrying the information, and it gets copied into an RNA message to do the stuff and go ahead in the cell. So so that's the first thing. So it probably was RNA. But then to translate your question into that how did it happen? So we don't 100% know for sure, but there are some very good ideas to do with that there were these. So the RNA chemical, a bit like DNA, can get copied. And so this copying could happen, and then it was probably originally in something not inside a cell. So this is just a molecule, and it's a chemical reaction. You're getting more of this molecule, and then at some point they start getting enclosed in a space. So one idea that I've heard at least Nick Lane from UCL talk about is the idea there might have been something like, you can imagine even little dimples in a rock, that it would be a little enclosed space rather than the wide open ocean kind of thing. So you got these enclosed spaces, it starts going like that, and eventually this enclosed space becoming a little cellular membrane, and then you start having things that look more like cells, and things that looks a bit more like biological life as we think of it now. But the idea is then, this is the idea. This idea makes sense to me in a superficial sense, at least like that. It's not what I'm working on every day, but that you have this molecule that originated before you would necessarily call it life. And it possibly happened multiple times without it becoming a stable and successful lineage that led to the life we're talking about today.
Thank you. The lots more questions. We've got one from Jonathan here. Jonathan wants to know, did humans evolve to be susceptible to viruses so we could get genetic upgrades?
So that's a funny question. I wonder how Jonathan means that. Does he mean it as a joke or as a totally serious question? But I'll answer it like a totally serious question. So, so one thing, so of course viruses are big in the news at the moment, but there are other kinds of viruses that do integrate themselves into our genome. So when you get infected, the virus doesn't just hang around in the cell, but it actually integrates its genetic material into the genome of the host. That would be us if we're the ones infected. And when this happens, there's a certain chance that the cell that gets infected is actually a progenitor of a reproductive cell. So then that virus DNA can get passed on. And this has happened sufficiently that 8% of the human genome is viral in origin. And there are multiple very interesting cases where this viral DNA that was lying around the genome that eventually stops acting like viral DNA. It just is lying there, but it got recycled and became really important and interesting genes from a human point of view or from a mammalian point of view. So actually, it turns out that sometimes getting a viral infection in an evolutionary sense has been a kind of genetic upgrade, because it's given the raw material, it's left some raw material there that has been recycled and repurposed into other functions. But I wouldn't say that we have evolved in order for that to happen. It's just that it has happened, and it has had some, ultimately in the long run, some useful effects, you might say. But we didn't evolve to get viral infections. Viruses more likely evolved to be able to be good at infecting us, and we're fighting back all the time in terms of our immune system. If you look at the mammalian immune system, has tons and tons of genes. It's a complex immune system, and there's a lot of genes involved in the immune system, and they are some of the fastest evolving genes in the human genome and in other mammals as well. And that's because of what's sometimes called an arms race with the various pathogens, viruses included, that our immune system is evolving rapidly, and the viruses are also evolving rapidly. And it's that the immune system is any changes in the immune system genes tend to be advantageous because they allow recognition of diverse infections or pathogens. But then what happens on the other side is the virus, any changes that happen in the viruses that mean it's not recognized by the immune system are advantageous. So we're both constantly running. And it's sometimes described as Red Queen evolution, after the Red Queen in Alice Through the Looking Glass, because she says that it takes all the running you can do to stay in the same place. So in one way, things stay in the same place. If you take one level of distance, the immune system still recognizes the virus or whichever pathogen it is. But if you look at a different level of resolution, it's totally changed or significantly changed, because the DNA sequence on both sides will have changed a lot. But so that's, so we didn't evolve we haven't evolved in order to be infected by viruses, but there have been interesting evolutionary consequences of that.
There's a certain kind of somewhere dystopian stroke utopian science fiction novel, isn't there, about the creature that didn't survive because it could never be infected. It kind of feels very counter instinctual.
Yeah.
So, Jonathan, I hope that explained that. It certainly helped me a great deal. Brigger would like to know, recently read someone describe mammoths as a genetic disaster, and that's why they went extinct. Are you able to talk about this, please? What makes something a genetic disaster?
I didn't hear that. Poor mammoths. I don't know if that, I think, okay, so I'm not an expert on mammoths, so I apologize then. I'll say something. I won't talk too much about mammoths in case I get it wrong. But what I have heard is that mammoths essentially were victims of climate change. After the ice age ended and they were adapted to cold weather, and it got too hot for them and they didn't manage to, the climate changed faster than they could, which I think is a lesson for modern times as well, because this is a significant risk at the moment, that the climate is changing faster than adaptation of various animals might be able to keep up with. But I didn't hear them called a genetic disaster. If you wanted to ask me, okay, so I changed the question. If you wanted to ask me what do I think is the biggest genetic disaster, I would say the platypus, because the platypus seems to be weird in every way that it's possible to be weird. So not only is it a really strange looking animal that had the Victorian naturalists thinking was a hoax, but it's got such a strange genome, and even though it's a mammal, its sex chromosomes are not at all like the mammalian sex chromosomes. They're a bit more like bird sex chromosomes, and instead of just being like an X and a Y pair or an X and an X pair, they have a chain of sex chromosomes that the end of one matches up with the tip of the other, and the other end of that matches up with another one, and it's all really weird. So platypus is I think the most genetically and in every other way weird thing I would come up with.
But it's not a disaster, is it? Because it's still going. That's the thing, isn't it? That's
still going.
You become a genetic disaster if you've actually managed to survive for a while. It seems that you're more likely to become a genetic disaster than start off There was more platypus stuff in the news the other week, wasn't there? The platypus research is still going on. We've had loads of questions in suddenly. We often get that in the last 10 minutes. Then they all arrive. This from Suzanne Jones. Suzanne, would she'd like to know, what is the most exciting thing for you that is currently happening in genetics?
Oh my goodness, there's so many things. I think I'm really interested in a lot of the ancient DNA work. I find that really exciting. So, and one of the things that makes it, one of the aspects of it that's really interesting is, so what we've done and what we've been able to do for a while is you're looking at modern examples and you infer the past. But then when we were able to get ancient DNA, it was literally stepping back in time, and it's getting the DNA of one of these individuals who lived a long time ago. And then that's been really, really, really extraordinary. And not only individuals then, as the technology in terms of extracting DNA from really what seemed like very degraded samples, but being able to get the DNA out of them has meant that now we've got lots and lots of data from ancient samples and really old samples, and it's quite extraordinary. It really is stepping back in time, and that's fun. And that's really, I find that really exciting. And just also, I mean, I've so many different things. I think the work in understanding, I'm always fascinated with understanding the process of development. And I think that's something that we're understanding better and better all the time. So that process of how is it you can start with a single cell that doesn't apparently have an up and a down, even though it might have a little bit, and you get, a structure like us, and it reliably happens every time, and the arms and legs come out in the right place, and all of these kind of things, and that's extraordinary. And it seems impossibly complicated, and yet it works every time. And I find that really exciting as well. Like as we're understanding more and more about that.
Thank you. William would like to know: can CRISPR help with COVID?
Oh no. Unfortunately, okay, CRISPR couldn't help with, I'll rephrase. You wouldn't CRISPR humans. You wouldn't do that. You wouldn't change humans in order to try and make you resistant to COVID. But actually, you might use CRISPR in making a vaccine. That could be possible, because one thing they might do is a potential strategy a vaccine strategy is to have an attenuated version of the virus, so like a version of the virus, but one that doesn't really make you sick, so that it educates your immune system without the illness. And that is somewhere that maybe CRISPR could come in, so that you could take the actual SARS-CoV-2 and modify it sufficiently that it still elicits the immune response without making you sick. So it could come in that angle. So I said no too quickly initially.
This one is someone who wants to program their own creature and grow them. So they want to know when that technology, when will there be, or how long until a Gene++ compiler comes out and I can program my own creatures?
Well, you possibly could do it already, actually. There is such a thing as there are people who do DIY biology, like I think DIY bio, and there's a lot of stuff you could kind of do at home in your garage. You do need, if you want to do any kind of changes, it's called genetic engineering or genetic modification. You do need a license for that, but you can get it. I know a guy who does. He's really big into doing the DIY bio and has all this stuff set up at home, and even made so 3D printing files, whatever you call those template files, so that to print the various components. Like, so you want a centrifuge, so he designed a little adapter for an ordinary household drill that you put on where the drill bit would be, and it spins and makes it into a centrifuge. And so he's got all these different things, and so there's things you could do at home, but you're not going to get a dinosaur. You might get a glow in the dark yeast cell or something. But that's fun if you get to do it, I suppose.
So it's quite close to that moment where people bought those what were they called? Not seahorses. Those little bits of shrimp that in the adverts in the, what were they?
The sea monkey, weren't they?
Sea monkeys. That's it. Yeah, that was.
It's not it's not completely inconceivable to me that there could be something a bit like a chemistry kit. That could be, a little genetics kit. It's not inconceivable that there could be some little things there that you could do maybe.
This is Malcolm would like to know. Now, this is I know it's a really tough question, and you may well want it, but it is that one. Do you think we'll ever be able to determine a percentage of environment versus genetics when it comes to behaviour? And I think that whole thing. I read Robert Plomin's Blueprint quite recently, and trying to understand those things and trying to, I suppose, ever actually have the situation where you are given the legal rights to create.
Yeah. So I'll first caveat is I don't work on that in the genetics of behavior, so I'll just give myself a little out there as well. But I mean, one of the things that is tricky to, I think it's a tricky concept in terms of when you talk about genes versus environment in any genetic trait, is that you're always talking about the variation in that trait rather than the trait itself, right? So let's just say that, I'll give kind of a stupid example of, if you were talking about what's the genetic component of how many legs you have, something like that. You would, if you look at it from this point of view of that we normally talk about in behavior, you'd say you wouldn't talk about a genetic component necessarily at all because there isn't really variation. Everybody has 2 legs. And so you're only talking about that bit that varies between people. So there's things that are really, really constant between people. And those are, let's say, highly genetic, but we don't start talking about this component. So that's the first bit. Then that gets tricky to understand. It's just that we are talking about the little variations usually. And so, but what happens is the amount that is genetic is down to your genes versus down to the environment can then vary depending on what happens in the environment as well. So you can get less variation, less environmental variation, perhaps. Like could be things like the schools being more standard and things like that, which means that all that's left is the genetic variation. And then it would look like it was more genetic trait than if the environment was itself more varied, and it could allow more of that variation to come about. So it's a tricky topic, but basically the answer somewhat depends. And I think it's always going to slightly depend. And even if it didn't somewhat depend, I think it's really, really difficult because behavior is one of those things that's notoriously difficult to quantify in a way that's amenable to scientific analysis as well. So it's not like you can just count it in the same way. You can count things and that makes them easy. If you have 3 and I have 4, I can say, there's a difference I can measure. But it's very hard to measure differences in behavior. I think there's certain things are the simple ones, right? So those are probably the ones people analyze first, but it's tricky. It's a hard question, and I don't know if I answered it necessarily very well. But
well, I don't think, as you said, it's not a simple. Then we'll get there, and this is what we discovered. As you said, it's that great thing. I think almost every time that we've done any shows where we've talked about that, it's always ended by saying it's become more complex. The blurred area in between has become blurrier and bigger. The information which does appear to be to some extent more rigid. Final question. This is a nice simple one because it's definitions.
How do I pronounce my name? No.
The Turtle Power would like to know, can you explain the difference between a molecular marker and a genetic marker?
Okay.
I was reading about this the other day, and it seems to be similar to how a square is also a rectangle, but a rectangle isn't also a square.
Okay, yes, it's probably that. I don't know. I think it might depend on the context, because they could be totally interchangeable. Yeah, like you say, a square and a rectangle. So a molecular marker. So essentially, a marker is just something you can measure. That's what a marker is. So it's anything that's amenable to being measured. And a molecular marker is usually going to be, it could be genetic in terms of it being a DNA marker, or it could be something produced in the cell, perhaps. But a genetic marker might be a trait that you're looking at, which could be a bit more perhaps than a molecular marker, but I'm not quite sure actually what distinction is being made there. It's not. It might depend on the context it was written about, but yeah. So, but they are almost the same thing, and I might need to see the context to be sure what they mean actually in terms of what distinction was being made there.
Well, that will we will leave it there and say that there's loads of stuff that people can catch up with in terms of, I said, all the electric picnic stuff there. The Christmas lectures are still definitely worth returning to. I mean, that must have been so much fun, and it must have been very interesting to see again in terms of having a large audience, which were, you know, a lot of children watching it, and the things you must have found there were certain things that arrived in your mailbox where you're like going, oh well, this one is really kind of, you know, sometimes even kicked up a stink, or sometimes just kicked up fascination.
Yeah, oh no, it was so much fun. I loved doing that, and it was great doing it with Alice Roberts because we just got along so well when we were doing it as well. I think we really had a laugh, and the audience, it was just the best audience ever, right? Because they're, you know, it's a whole group of people who've chosen to be there, and they're there because they're really enthusiastic, and they're practically on the edge of their seats listening to you, which is really nice, right? It's nice to talk to a group of people who really want to hear you. So it was so much fun, and coming up, you know, finding examples to talk about there. And the Christmas lectures, of course, are really big into visuals and what they call demos, you know, things that you can wheel in and mess around with and make pop and do all these kind of things. So getting creative about that in a way that I hadn't done before, because I don't normally work with props, you know. So that was fun. It was really, really good fun. It was definitely a wonderful experience. I'm delighted I got to do that.
They should definitely at some point show that room, that Victorian room where everyone is held, all the people who are doing demos. So you've got a man with a bunch of crows, and you've got three people dressed up as chimpanzees who are on stilts, and you've got a great big vat of liquid nitrogen, and it's a proper circus sideshow of science, isn't it, when you actually see behind the scenes.
In one of the lectures we had dogs coming in because we were talking about all of this big genetic variation within this one species, and in that room had a sign, you know, like, you know, don't open the doors, don't let the dogs out. There was a pile of dogs in there, of course, from something like a Great Dane down to I think the smallest one was a little Chihuahua that I carried onto the stage, and it was trembling. So the poor little thing, its owner was just a few steps behind me. But it's funny. It's the whole thing, and we brought in a big horse, and a cow, and you know, all kinds of things. The lift was well used in that place to get all these things in, all those weird animals. It was fun.
I remember Mark Miodovnik perhaps not having fully checked the height of the door when he rode in on his penny farthing. Thank you very much, everyone, for watching, and all the previous episodes are online. You can go to Genetics Unzipped. You find them on Cosmic Shambles. Thank you very much to the Genetics Society, the Milner Centre for Evolution at the University of Bath. As I said, we have one more full Genetic Shambles in 2 weeks' time with Steve Jones, author of one of my favourite books, actually one of my favourite science books, Almost Like a Whale, which changed the perspective of so many things for me as a non-scientist, and I think also scientists. Well, there will be many stories on that, and also I should mention Helen Chersky and I will be doing our Sunday Science Q&A this Sunday live, and Helen, the reason I mention that is because Helen is one of the people doing this year's Christmas lectures, which should be very exciting as well. Thanks very much to producer Trent Burton. Enjoy the rest of your week, and bye-bye.